So I will be quite brief with regard to the diversity. I think this has been marvelously presented by the speaker before me. This shows you just the incredible complexity of the microbiome as we know today. And as has been highlighted before, I think we are more microbiome than human beings. The question for me was always, who is the host and who is the guest in that whole game? So he also mentioned that we have to be very careful in the interpretation of many microbiological results because this is highly depending on the localization. And I tried to compare this a little bit like kind of climatic areas, which are different from the face to the dry areas to other face, to other localization on the skin. So all these different localizations harbor a different kind of composition of microbiome. Something which has not been mentioned, which is highly interesting to me, is the relationship or the role of the microbiome with the body oral. I think you always have been in this kind of strange situation where you met somebody and you immediately, beside talking with this person, had the feeling the chemistry does not fit. In French, we say, or in German, we say, ich kann ihn nicht riechen. And this probably comes partly from the microbiome, because we now know that parts of the substances which are produced, particularly in the axillary area, in fact are metabolized by the microbiome and are generating particular kind of quite probably not really perceptible smell and signatures that may or may not dictate your own behavior in presence to this person. Another thing is that for mosquitoes, this could be very appealing or not. You probably had the same kind of experience that you are lying in the bed with your partner and you're always claiming that you are always the target of mosquitoes while your partner just lying beside you has never been stung. It's probably explained by your microbiome. So it's all about dysbiosis on the skin, and this has been already discussed before. We now better understand what is the role of the microbiome, what is the role of that kind of crosstalk between the different members of the microbiome and the skin immune system. One particular example of dysbiosis, which may be relevant for our understanding of the disorder, at least partly, of course, is atopic dermatitis. And this is a typical picture that you see in your clinics with a small kid with a quite crusty erosion and lesions, typically super infected and probably impetigo-like atopic dermatitis lesions. And what we know currently is that this crosstalk is extremely important to understand between, I would say, the good guys and the bad guys. The good guys are the commensal, particularly Staphylococcus hominis or epidermidis, and on the head, Staphylococcus aureus. So there is a constant crosstalk between these different kind of bags on the skin. The quorum sensum is something which is explaining this kind of interaction. And what we also have learned over the last years that there must be something in the skin immune system which is translating the information of these different bags on the surface or in the skin to the immune system, particularly to the adaptive immune system. And we have found in our group in Bonn that the Langerhans cells are sensing the presence of the different kind of bugs on the skin and are transmitting the information to the adaptive immune system, and that in atopic dermatitis, this sensing is in fact not working. So the Langerhans cells are tolerized, more or less, to the Staphylococcus aureus derived toxins and signals and cannot, let's say, transmit the information anymore to the adaptive immune system. And this could also partly explain why you have much less Th17 cells, which are helpful in order to fight these bugs on the skin in atopic dermatitis compared to other skin disorders. Another point which is interesting in terms of understanding the role of these bugs in atopic dermatitis is the very nice correlation between, on one hand, the biomarkers that we can measure in the blood, particularly IL-13, which is, as you know, probably now is the main driving force in terms of inflammation in the skin and atopic dermatitis, but also other cytokines like IL-22 and of course TARC, which is one of the best biomarker in terms of correlation between severity and biomarkers in the blood. And we know that when you are now using these new kind of targeted therapies, dupilumab, tralokinumab, for example, you have an impact on the composition of the microbiome. There is no discussion about this. This has been nicely shown, particularly here, for example, with tralokinumab, after 16 weeks in the first of the 2 pivotal studies, EXTRA-1. You see here a very strong impact with the reduction of Staphylococcus aureus colonization in the skin of these individuals. The same holds true for the other one, dupilumab, which is targeting the alpha-1 chain of the IL-4 receptors, and you have exactly the same phenomenon. But this does not mean that these treatments are directly affecting the bugs. Most probably, the effect is indirect. Most probably, in fact, the reduction of the inflammation is reducing, let's say, the culture conditions for this Staph aureus, and by this indirectly is reducing the colonization. But beside this, there are a number of other strategies, particularly those that have just been mentioned, trying to modulate, to intervene at the level of the microbiome itself. So there are many different kind of attempts currently, and a lot of clinical trials running, trying to show that impacting on the Staphylococcus aureus colonization, you may indeed have a short but also a long-term effect on that colonization. So this is just an example from a paper from the group of Rich Gallo. They have used different kind of extracts, particularly the bacteriotherapy using that Staphylococcus hominis A9, which seems to be a very interesting approach in order to substantiate or to, how to say, to introduce in a forced manner this kind of good guy and to replace the bad guys by the good guys. A very simple kind of explanation. It seems to work. And you see here a lot of other approaches that are currently in the pipeline trying to have an impact and to modulate the dysbiosis in this condition. And this is what I just mentioned. This is this Nature paper from the group of Rich Gallo, which have nicely shown that this particular kind of bacteria is able to kill the activity of Staph aureus, but also, and this is interesting, on the other hand, to modulate the inflammatory effect that is measured on site. And as hoped, there is no side effects seen in this clinical trial, so this means that the trial will probably go into the next phase of development. Overall, when we consider the pathomechanisms behind atopic dermatitis, I think we have now to realize that there is no single one reaction, which is now commonly described as the T2 reaction. We now know that there are a number of different kinds of reactions, and I think that the microbiome has a playful role in this disorder, but in my particular understanding of the disorder, it probably plays a role at the very beginning, at the early phase, what I call the phase 0, the phase 1 of the disorder. That is, in conjunction with the activation of the innate immune response locally, which starts with a, would say, an allergen, unspecific, independent kind of unspecific inflammatory reaction at the very beginning of the disorder. In that phase, the dysbiosis most probably has a very important role to play in the deviation of the immune response into the direction of Th2 response. And that's the reason why I think most of the approaches that we will see or see currently in the development in terms of modulation of the microbiome should concentrate, should focus on that very, very early phase of the disorder, because this is most probably the critical window of opportunity for this kind of intervention. And last but not least, the Holy Grail, the big question, is still not answered. What was first? Was it first the inflammation, which is inducing the dysbiosis, or was this the dysbiosis which is inducing the inflammation? this question is still not answered. And we need much more data in order to understand what is going here. So finally, I think we have learned a lot about the microbiome. Another hype, I would say, since more than 10 years now. But a hype which has hidden a lot of hopes. And these hopes, I think, can now be concentrated in the development of new approaches. What you have seen here with endobioma is clearly a revolution in my opinion. But there are many other approaches that are going in the same direction, trying to have an impact on the disorder. And for me, and for you as clinicians, the most important thing is not to have the control over the inflammation on the short term. The most important and challenging aspect in this particular disorder is to control the disorder on the long run. This is the real challenge, and we hope that by manipulating in the right way the microbiome in the future, we will be somewhere nearest to that goal. Thank you very much for your attention.